Thank you for joining Insights and Discoveries with Metabolon webinar. I hope everybody is doing fine and keeping safe. My name is Paweł Gardzielewski and I'm joined today by Soumya Shetty and Vinay Pawar. We will address some common questions we receive from researchers about metabolomics and how it can better their science. The presentation will be muted, but we welcome questions that can be answered during the questions and answer session at the end. To submit a question, please open the Q&A window at the bottom of your screen. If you have technical questions, please use the chat feature in Zoom, also at the bottom of your screen. We will feature a poll at the end of the event and welcome your feedback. So, to begin, I would like to briefly introduce our company. Metabolon's headquarters is based on the East Coast of the US in Research Triangle Park, North Carolina, but we have a global presence with analytical testing sites worldwide. To date, we have over 200 employees, with many of them being data scientists, biochemists, bioinformaticians, generating and processing the data. Over the last 20 years, we've pioneered the field of metabolomics, establishing ourselves as the world leader in the field. Today, Vinay and I will address key questions about benefits of metabolomics, guidelines on study design and sample preparation. What type of metabolomics analysis to use, depending on the objective, and what the results of metabolomics experiment look like. We'll also share some relatable case studies that hopefully will help you visualize what your project could look like. Vinay, many of our customers, or should I say prospective customers, want to understand the benefits of metabolomics versus other modalities. What do you say when asked about that?
Yeah, we typically communicate that a benefit is for doing metabolomics is that it's being closest to the phenotype. So genomics, can provide you potential risk associated with that particular gene. And while proteomics can inform you about presence of a particular protein, but not necessarily what the function is. In metabolomics, we will be able to reveal what is really happening and what is the active biology behind that particular state and understand the disease in detail.
Very well indeed. So once somebody is decided to run a project with us, they want to know how to set things up. I get this question often. How do I set my experiment? How do I collect the samples? And how stable are metabolites in my sample?
Yeah, most metabolites are stable once segregated from the adjacent enzymes. Clients can help ensure stability by harvesting samples as quickly as possible and also storing them immediately at minus 80 degrees Celsius. Metabolites are predominantly very stable at minus 80, and only a small subset is unstable at minus 20 degrees. We have covered a number of studies over the years, and a good example to illustrate stability of metabolites in terms of storage can be found in the JIVA studies, which we have done in past. So these JIVA studies, they span to over 5 to 10 years of period, and we were able to elucidate the association of genetic data and from two very large independent cohorts and individuals with metabolic signatures. So several very strong associations were identified, and metabolite levels were and various SNPs supporting the idea that metabolites can be very stable and useful as a biomarker of disease. In terms of experimental design for metabolomics, it is always ideal to have a systematic protocol as much as possible. While X will impact the levels of metabolites among the test subject and can complicate the biological interpretation, undesired influence on underlying biochemistry, for example, these things can be minimized through study design and proper study power. So based upon our experience and we have established certain guidelines. We provide also a sample collection kit, and we also recommend optimal concentrations from cell tissues, and also provide a proper sample collection guidelines. Typically, I usually recommend for a good study design is to, if it's a cell line experiment, then usually you should have 4 to 7 replicates. If it's a small animal experiment, then it should be from 6 to 10 in each group. If it's a large animal, then between 8 and 15. If it's a human study, then I would recommend to have 25 to 40 subjects per group. So our PhD level biochemists will discuss the ways to mitigate the impact on your specific needs and study goals, and that's the best way to design a particular study for metabolomics analysis.
Very well, thank you. I want to remind everybody, if you scroll down on your screen, you can see the Q&A feature. Please type in your questions there and we'll answer them at the end of this webinar. Okay, so once a customer can picture a project with Metabolon, they want to see how their data is going to be reported. What can we offer here, Vinay?
Yeah, so typically the data analysis is at Metabolon is conducted by a PhD level biochemist using their expertise and knowledge and the literature available, along with the bioinformatics tools to interpret biological relevance of observed fold changes in the metabolites across different matrices, different time points, conditions, and doses. So this is usually displayed in heat maps and bar graphs in an Excel. In addition to that, we provide these data via our My Metabolon client portal, where you can download the study deliverables such as a Word doc, which is an interpretation report, and an Excel sheet. The My Metabolon client portal also provides you data analysis and visualization tool possibilities that allows for further data exploration. And there you can visualize different firework plots and really interact with your data in detail. In my personal opinion, one of the key advantages to work with Metabolon is your results are always leveraged against Metabolon biochemical knowledge base, which has been developed over the past 20 years. We have analyzed more than 500 different diseases and really gathered on insights into all different variety of matrices. So this is all can be leveraged by working with Metabolon.
Very good. Okay, so this was the global panel Metabolomics. But that's not all that we offer. In which scenario a client should choose targeted analysis over untargeted analysis?
Yeah, that's a very good question. So Metabolomics, particularly our global and targeted HD4 platform, is a discovery tool, which serves the entire Metabolon for gaining biological insights and discovering biomarkers. It also guides you in the direction of in the direction your research you want to take, which you should take. So untargeted analysis is very good for hypothesis generation or biomarker and target discovery purpose. Like any research, it needs validation. So many clients follow up, using targeted assays, our technology services. Many clients choose to conduct more studies with us and tackle the follow-up experimental designs or ask other research questions.
And could you briefly tell us what are the Metabolon targeted offerings?
Sure. Our targeted assays are specific metabolites or signature of metabolites, which can be, track specific biomarkers and improve the biological understanding for both preclinical and clinical research. They are excellent tools for rapidly, and accurately establishing, let's say, PK/PD studies and translational studies, understanding efficacy of a drug or a safety of a drug in a clinical trial. Also, these target analysis could be, used for post-market surveillance studies. I think there is a unique advantage of using metabolomic assays over, legacy assays such as ELISA or IHC. These assays can be, used on your needs and develop and validate for your, let's say, research-only purpose or GCP purposes, or also, clinical laboratory improvement amendment purposes. Some of the off-shelf offerings we have is, let's say, we have a fatty acid metabolism panel, which measures the key fatty acids important for human, animal nutrition and understanding metabolic disorders. There is a short-chain fatty acid panel, which can be, helpful in understanding importance of gut microbiome on human and animal health. In addition, we have an amino acid panel, we have seen wide use in variety of genetic disorders and acquired conditions that influence this amino acid metabolism over the period. And similarly, I have seen extensive use of amino acid panel as a useful tool in bioprocessing, cell culture applications.
Very good. And with the targeted panels, can we detect a particular lipid species like phospholipids or tags, triacylglycerides?
Yeah, sure. So we actually capture a robust representation of lipid metabolites. While we do not detect the tags in our HD4 platform, but we do detect our range of complex lipid. The coverage is not comprehensive as the complex lipid panel, but for most of the studies, we can gain a good understanding of membrane biochemical diagnosis, lipogenesis, lipolysis, and fatty acid oxidation through our complex lipid panel.
That's interesting. Okay, let's try to put this a little bit more in the context from customer point of view. Can you share an example where we helped with biomarker development?
Yeah, sure. There are a number of examples, particularly which comes to my mind is the GWAS studies, the population health studies we have done with the Department of Human Genetics in University of Texas and Sanger Institute. We found in that study that the genome-wide significant associations at 45 metabolic loci and their biochemical connectivity to 400 metabolites in human blood. We extensively characterize, the results and the in vivo blueprint of human metabolism, and with that information on gene expression, heritability, and the overlap we known for loci with complex disorders, for example, inborn errors of metabolism or pharmacological targets. Metabolomics identifies, genes and metabolite causal. For, let's say, high blood pressure, hexadecanide, one such metabolite, which was highly associated with these blood pressure phenotype in 3 different cohorts and shown to be as a causal. So identification of a novel target was achieved in that, and avenues to gene function and understanding disease etiology by integrating, different omics technology into deeply phenotyped population. These are the biomarker work we have done in that.
Yes, I remember the HEXA study. And have you got an example how Metabolon helped in immuno-oncology research?
Yeah, sure. You probably know that the checkpoint inhibitors are the key focus in the current immuno-oncology research. Due to its, applications in treatment of various malignancies, CTLA-4 and PD-L1 are 2 most prominent targets for unleashing beneficial effects of immune system. To understand the biology of these targets more, in detail, some of our collaborators perform a system analysis with us, including metabolomics on T-cell targets with ligands for PD-1 and CTLA-4. We have helped them to understand the PD-1 and CTLA-4 associated distinctive metabolite programming in T cells. So as both, PD-1 and CTLA-4 stimulate stimulation results into suppression of glycolysis, and however, in some patients, we have also seen, particularly with PD1, stimulation results into increase in fatty acid beta oxidation. And these differences resulted in improved understanding of impaired T cell effector function, mediated by PD1 to provide, potential targets, and also biomarkers for overcoming and detecting T cell exhaustion pathways.
Very good example there, Vinay. You mentioned on one of your previous slides Metabolon's compound library. What is it and why is it important?
Yeah, our library is built around the compounds related to mammalian, plant, and microorganism or metabolism. So our mass spectral methods mainly rely on, UPA, LCM, SMS, which offers, great sensitivity for many metabolites compared to, let's say, gas chromatography methods. Our library is most comprehensive in terms of, chemical spectral library to the date. We are able to cast a wide net in untargeted approach across broad sample types and populations. To the date, our library contains roughly about 5,200 small molecule and complex lipids. By far, the largest reported in the current scenario, providing more coverage and leveraging biochemical context through untargeted metabolomic studies. If you don't have a coverage, you are likely to miss a biomarker or pick up something that isn't sensitive or have a limited knowledge through a limited context of the changes which you see in the phenotype. If you are trying to uncover something new, you can't look into known database or literature. That's when the untargeted metabolomics with the highest coverage in the library makes much more sense. For complex use, such as immunotherapy, comprehensive coverage is crucial. Cast wide net of biomarkers related to immune and tumor functions, as well as more patient-centric signatures, like, for example, lifestyle, like the xenobiotic influences, diet influences, microbiome-derived compounds, all of which may modulate or relate the signature for response of a therapy. So this is all is present in our library. So when you do identify a compelling biomarker or set of biomarkers, we have a technical capability and experience, capacity with you developing a custom targeted panel in the end. That way you can track those molecules through the drug development or product development in the clinical space and for GCP validation if necessary.
Absolutely. So in essence, you give yourself the best chance to succeed by taking advantage of the wealth of the library that's there. And lastly, do you remember a case where we exceeded customers' expectation, where we provided novel insights that perhaps were not anticipated beforehand?
Yeah, I can definitely speak of many examples of this. Most clients, are impressed by the amount of data and the insights we provide through our interpretation reports. Examples, might include identifying, let's say, mechanisms of actions, mode of actions, which was completely different than what was expected from that particular drug target. And also determining the off-target effects of that particular compound and drug. Identifying, biomarkers to improve performances, let's say in bioprocessing, in cell culture, and fermentation. The ability also to stratify, degrees of diseases over the periods, especially in terms of chronic diseases. So this is where we have, exceeded the expectations from the customers, and they were really blown up by the analysis we provided.
That's great, Vinay. Thank you. I think we have some questions from the audience, Soumya. Over to you.
Thank you, Pavel. So I would like to thank Pawan, you and Vinay both for this informative session, and I hope the audience is enjoying it. So I would actually like to request the audience to use our poll feature to provide their feedback so that we can improve their experience and also work on future webinars. And with that, I would like to switch tables now, and our panelists would be answering questions sent by the audience. So we have already received quite a few questions, but I would still request the audience to use the Q&A feature and send their questions. And we will try to answer them all. So moving to the questions, so I mean, in the Corona situation, we have received perfect questions. So I mean, asking you, Vinay and Pavel, so how do we work with infectious diseases such as COVID-19 in the current situation?
Yeah, yes, indeed. Our main lab in Arkansas, North Carolina can work with, biosafety level 2 and below agents. In the past, we have been involved in several HIV and similar viral infection projects. Currently, we are involved in several COVID-19 projects in European Union and also with the US clients and collaborators. Due to urgency of the matter, we are offering, faster turnaround time for those projects and then giving priority to those samples for analysis on our platform.
Thank you, Vinay. I think another related question to this, I think because of the ease of sending samples. So are we able to analyze, metabolites by using dry blood samples?
Yeah, that's a good question. So yes, we do analyze dry blood spots. We are able to do complete metabolomic analysis on that. This is, this has been successfully done in several projects, particularly in inboard areas of metabolism studies.
Great, thank you. One more interesting question. How do you see the potential application of metabolomics in forensic analysis? Which are the main limitations?
Yeah, in terms of forensic analysis, it really depends on which sample types it is, because LC-MS is a very sensitive method. So we are able to provide a key, let's say, causality and the agents which are cause for a particular phenotypic condition. So of course, this is one of the key areas we have been also providing our services.
Great. So another good question is, what type of metabolite does Metabolon measure?
Metabolites include particularly, amino acids, small peptides, sugars, many types of lipids, nucleotides, including, precursors and degradation products, and diabetic compounds, drug and drug metabolites, xenobiotics. For a good reflection of, classes covered, I would recommend you to contact us. We can provide you some of our brochures and give you all the classes of metabolites, information about all the classes of metabolites we detect. But I would like to highlight here the number of metabolites detected depends on the type and the complexity of that particular sample on the matrix. For example, in human plasma, we often detect in the range of 1,000 metabolites. And for feces, for example, we can detect roughly up to 750. For cell culture media, I think we can detect up to 600 to 800 metabolites.
Great. I hope you got the answer. So guys, I would like to request to send more questions, and if you have any further questions on the questions asked, feel free to reach out to us. In the meanwhile, so next question for you both is like to explain about the mode how we develop our libraries. And except of purchasing analytical standards and analyzing them on machines, are we also synthesizing compounds? How do we achieve level 1 of annotation for completely new biomarkers? I think multiple questions packed in one question. So go ahead, guys.
Yeah. So I think we have developed our library for over past 20 years. So we have optimized our methods and protocols accordingly in past 20 years. So we typically we typically use 4 different detection methods, which is based on these are non-polar or polar molecules, if they are lipidic in nature. And on top of that, I think we use a tier 1 identification approach for identifying metabolites. So for example, what we do is we not only look at the absolute mass, but also retention time and fragmentation patterns. So when these 3 key things meet the criteria, then that's our tier 1 identification approach for metabolites detected in a particular sample. In terms of standards, most of the standards are synthesized in-house, and some of them are commercially bought. So that's the kind of approach we have used.
Great. And an overlapping question, Vinay, is how do we extract our samples?
Oh yeah, we mainly use methanol extraction in 80-20 ratio. Methanol extraction has been found to be most effective and straightforward. Reproducing it's reproducible for our global metabolomics analysis. It can extract and recover compounds from diverse metabolic classes. The method has been broadly used in metabolomics field.
Great. So next question, what is the impact of sample degradation on metabolomics results? How rapidly after collection should the samples be analyzed?
Yeah, I think what is important to have proper study is to treat your samples all in the same manner. So the metabolites are pretty much stable for, let's say, 1 to 2 hours after collection of samples. So the important thing is you should treat your all group in the same manner and then the store in the same manner, because that would avoid any bias creating because of the sample handling.
Thank you. So moving on to the next one, do you run metabolomic analysis from prostate cancer cell-derived organoids?
Yeah, that's a good question. So we have done similar studies. I'm not so sure if you have done it in recent time. I just have to cross-check if you have worked with organoids derived from prostate gland. But I think we have worked on organoids and several other vesicle projects.
Okay. So another one, Vinay, is, do big proteins interfere with our analysis? And if they do, what kind of impact do they have?
No. So for our extraction method and detection method is optimized for only small molecules. So we do not see any interference coming from large proteins in our measurements. Particularly, our detections, the small molecule detection is up to 1,500 Dalton.
Thank you. Audience, yeah, we have almost answered all your questions. So if you have more questions, feel free to send it to us. I think we'll, wait for 1-2 minutes to take more questions. And if you have, more specific questions that you would like to be answered one-to-one, you can reach out to the team later. But if you have more questions that we can answer now, please send it to us. And maybe in the meanwhile, Vinay and Pavel, if we can share about like how we are managing things and how our lab is still running during Corona situation, if we can, like share with our audience, that would be great.
Maybe I can speak to this a little bit. So we have been granted the key status by the US government, and we are operating maybe not as normal, but we are ready to receive the samples. If you work with COVID-19, that's not a problem. And if you're not working on COVID-19, even more so. Our operation is, like I said, a bit scaled down, but the lab and the data analysis is ongoing. We're open for business. We're ready to discuss projects, and we can process them in a timely manner. What one can anticipate is that in time to come, we will see an increased influx of projects which can have an impact on the turnaround time. So securing your project now will ensure that you receive your data as soon as possible. Vinay, would you like to add anything?
No, as I said earlier, that we involved in several COVID-19 projects. We are considered as one of the essential services in US. So I think it's a need of the hour. Everybody has to contribute. So playing our part with our technology.
Thank you both. So yeah, we as I said, I haven't received any additional questions yet, but as said before, if you have more specific questions or if you have any questions that we can answer for you now or in the near future, please feel free to send it to us. And I would like to thank all of you for taking time today from your busy schedules and being on this call and participating in this webinar. So have a great day, and we will connect with you with your answers soon. Thank you.
Thank you.